It's a great pleasure to speak here. I dedicate the lecture to my colleague and friend George Ola, on whom you heard just a few talks ago. And I will try in this short time to discuss several aspects of modeling complex molecules by computer simulation. Okay, so in general, a system that we already heard about and we are interested to know how it works is the cell, because once you know how the cell works, you know how the body works. We have now a project about pancreatic cell where my co-worker tried to describe the cell as a city where you have things coming in, coming out, and going between different places. You see it better. Start the movie, please. In this movie, where you see example of a transaction of a signal from the cell surface toward the nucleus. And it looks clearly very complex, and it should look complex. And if you try to understand how it works, of course, there are network descriptions, a lot of experiments. But if you try to understand how it works on a molecular level, you have either to have this type of boat and to swim into the cell and see everything, or develop a physical model which will look at each element of the cell and try to understand how it works. And the cell composed of proteins that transfer the information. These proteins do different type of works. A lot of them are enzymes which perform chemical reactions and then are being put on or off by other enzymes. So we will discuss a little how we model the action of enzymes. This will be about half of the talk. And then what we do with more complex molecules that do different function in the cell. So for those of you who don't know, enzyme is a protein composed of amino acids. It binds other molecules called substrates, and it either break them or connect them. And the main point that it does it much, much faster than the same process without the enzyme. So enzyme is a catalyst. And for example, here we see what the enzyme is doing. Below, it's the enzyme working, and above is the same chemistry, but in water. So the Pac-Man represents here the enzyme. He tried to cleave the molecule. It could be in your digestive system. And above is the same process without the help of the enzyme. So it does its stuff very fast. And we want to understand how this is happening, and maybe more importantly, why it's happening so rapidly. And we want to understand it based on the structure, which is given by either X-ray and sometimes NMR. Another type of problem that should interest you from intellectual point of view are molecules that work like motors, like this F1 ATPase. It's a molecule that use the energy of the cleavage of ATP, it's the source of energy in many processes in the body, to rotate and to do work. The structure is known. We want to understand why it rotates to one direction and how does it work. And as I will argue, it's very hard to understand it just by looking at the structure or even by regular experiments. So, this is what we understand very easily. Could you activate this movie? Okay. Could you activate this movie? The answer. So this we understand. We want to understand on the same level how our motor works. And understanding is not simple, like when you watch a clock, you do not necessarily, unless you are a real expert, understand how it works. So we want to understand on a level that a watchmaker understands the watch. If you are a physics major, you know this exercise, you pour water, and you know how much work will happen. We want to gain the same understanding for our ATPs. Now, some understanding exists now. Essentially, they gave Nobel Prize for molecular motors, where people construct them and show that they work in one direction. It is not clear that the people who construct them fully understood how, let's say, ATPs is working. So we want to understand how the biological model works. It might also help us to design better molecular motors. So we have now two parts to go. One is to talk a little about how enzymes work, and perhaps how to design artificial enzymes. And the second is how to look at molecular motors and to understand them. By the way, this is how some people describe the action of ATPs, essentially a lot of people. You have this car. When you go down, you push the brake, so you could go up without moving backward. And then when it goes down, you release it. By the way, when it moves back, it's just like continuing the movie. So try to think about it as only moving forward. So here is the idea that you move just by the vibrations, the Brownian motions of the system. A lot of people like it because it reads them from thinking how it works. And I present a question to you. Is this story real? If it is real, why? Okay, so now so we stop here, we will come back to ATPs. We want to model enzymes. So, just to remind you, enzymes are proteins. Proteins are built of amino acid. You want to see how they are working. It's very hard to do it just by experiments because it's very hard to see what are the individual forces and so on. So a beginning in this direction is to represent the atoms and their connections as ball and springs. And it's essentially by this type of functions, and these descriptions appear to be very powerful. Essentially, most molecular dynamics programs are based on such description. It helps, this is the same story, so I'll skip it. A lot of these ideas were developed at the late 60s with Mike Levitt and Schneur Lifson. And they show you how to describe a molecule without chemistry. So you could describe it this way: a bond works very nice, but when you want to describe chemistry, breaking bond, you cannot do it as ball and spring. So you need to do something extra, and one very important direction was to describe the chemical part by quantum mechanics and the rest by classical force field, which allow you to treat the chemistry of the bond breaking in a way that allow the electrons to move, and the rest of the environment to include in a simpler way. Now, with this approach and modifications that I don't have time to go into, we could ask how molecules in the cell are working. One example, I will give you very few examples, is this molecule Ras P21, which has, I'm just trying to see if, this molecule has 3-phosphate GTP, and when it has GTP, the cell is not differentiating. When another protein called GAP is coming, this enzyme becomes a good catalyst, and it dissociates to GDP, and then the cell is being told to continue. So you need the extra structure. These are the phosphate of the GTP. You want to model it. Very complicated. This we switch. So you have to model the chemistry of phosphate hydrolysis, which is hard to determine experimentally because you have very high energy transition state. A lot of experimental work, but the only way to know what is happening is to model first this reaction in solution. Could we do the movie? This is a very expensive simulation of the chemistry of breaking this GTP in solution. And after you analyze the simulation, you could generate this very complicated energy surface. In using them, you could move to the protein. Activate this movie, please. No. Activate any of these movies. Yes, so this is this is a water molecule attacking the phosphate. This is the magnesium, and the bond is breaking. So you could produce nice movies, and more importantly, calculate the difference of doing the chemistry in water to doing it in, for example, GTP. This is a long story, I'm not going to talk about it, but you could figure out how these proteins are working and being activated. You could also figure out why enzymes have such a strong catalytic power by trying to simulate many enzymes, figuring out different suggestions, finding that most of them are incorrect, and then realizing that the enzyme works so fast because of electrostatic effects, which again, I'm not going to discuss. But we could reproduce the effect of the enzymes moving from solution, which are the blue lines, to the enzyme active site. Which means we could understand how enzymes, where we know the structure of the enzyme and the substrate, are working. Now the question is, could we use this knowledge to build new enzymes? Because people seem to believe you more if you generate new enzymes. So we could take the computer and change different amino acids from one to the other. We were doing it long time ago, so this is from the time where this Ninja Warrior was popular. And when you do it, you could take, for example, Christmas mutase and determine the catalytic power. These are different mutations. You could do it on another enzyme, so we can predict the effect of mutations. The question is, could we then take our knowledge and build new enzymes? And it's easier to talk about it than to do it. So we try for the next 5 years to work on CAMP eliminase. This is this enzyme with a CAMP reaction. And we were able to reproduce the effect of directed evolution. Again, not to predict, but to take the computer and to get the results. Looks very encouraging, but when we try to understand why directed evolution, which is not following regular evolution, works, we found something very strange. We found that the designer enzymes, instead of being really smart in stabilizing the negative charge that is formed on the substrate, where is the substrate? Here, during the reaction, it does something which I keep teaching that should never happen. It's destabilize the ground state. No enzymes that we know work by destabilizing the ground state, yet directed evolution does it. So it was very annoying. And fortunately, 2 years ago, Don Hilbert continued to play with this enzyme, and suddenly he finds that if you continue evolution, it works by stabilizing the transition state. This was very nice, and we tried to do even better. And then we found something which is very disappointing and create a major challenge to real enzyme designers, which means people who try to do it by being fully predictive. We look on his evolution and found that the groups that stabilize the transition state, let's try to find it, this glutamine exists there in earlier step of the evolution. But it starts to work only in the last steps where other group place it at the right direction. Now this means that for computer-aided enzyme design, it is not enough to figure out all the possible single mutations. You must do at least 2 mutations to figure out what to do. We found it about half a year ago. We have to fight the editor to explain that this is important, but it presents a major challenge. Okay, so now I will move to much easier issue, which is the ATPase and other motors. So let's say that we want to model more complex system. This very large molecule ATPase or GPCR or something else. Now, if we have small system, we could model it by regular molecular dynamics. If we have large one, there are 2 options. One is to wait until this specialized D-Shore machine will be able to really do it, or to use tricks, including using Brownian dynamics. So one trick that we use, which go back to 1975 with Mike Levitt, is to simplify the side chains to spheres. We also add electrostatic improvement, which I will not talk about. And then we use Brownian dynamics, this fish, instead of full atomistic dynamics. With these approaches, we could explore really large systems. And the first one I describe is this, could you apply the movie? Here. This is ATPRs. It has this central shaft. And when it rotates, when the chemistry occurs, they rotate this shaft and apply torque on whatever connected to it. Another interesting point, which was found by single molecule spectroscopy, if you connect beads to it, you could actually see the rotation. So it rotates, kind of randomly, but with a direction, and it rotates in a strange way. It does not do 120 degrees. It stops first at 80 degrees. Very nice puzzle that you have to solve with the structure. So we take our simplified protein model, augmented by electrostatic treatment, and we find this very strange and essential valley where it does not start to move to the chemical direction before it reaches 80 degrees. And with this system, we could build the landscape for this motor, run dynamics on it, I will not talk about it, and get a description which is similar to what you already saw. Apply the movie, please. Or maybe I will apply the movie. Yes. So we could describe how it works, and we could also describe why it goes in one direction. I don't have time to explain, but it's really require not only to use the chemistry of the hydrolysis, but also to give the right directionality, where going to the left has lower energy than going to the right. I will talk on it in my next example, so you will really understand it. I'm showing it back. We will agree that people who figure out why this car works could send me email. And I will show you one more example. We have enough time to do it slowly. Now, this example encompasses both directionality and energetics for a motor. So we do this now slowly. This is myosin 5. It's a molecule that has two heads. It carries load in the cell, and it does it in one direction. There are a whole family of myosin, including those that activate your muscles. In all of them, there are different views how it works. Most people don't really get it. They believe that the Brownian motion is what drives the motor. There is a concept of power stroke, very popular, which we'll see if I have time to show that it's incorrect, but it's basically saying that because, okay, first I will show you the motion. A movie, please. This guy moves in one direction. You see, when this arm moves, some people believe that because the arm moves, it pushes itself in one direction. And there are too few people who are brave enough to ask them. I don't see why it has one direction, because actually the chance to move here or here is equal. I mean, this tilt does not exist in reality. So let's define the question again. Usually apply the movie, please. And maybe try to put the microphone near the computers. Okay, usually there is a movie. So this what happened, stronger? Okay, this what happened when you usually walk without, same way to both directions. But in myosine, it's like one of them has problem walking. So it walks in one direction, and it's a very fundamental question, why this is so. So it's not easy to see from this picture, but if you see the two heads, you will find that there is absolutely equal tendency to go in both directions just from the cycle. So we work on it like since from 2013. We found some asymmetry. We found that one structure, when it is like this, has 11 kilocalorie lower energy than the other. And for a while we thought that this is the reason for the unidirectionality. We also drew, with Dean Eistman, we drew this type of 2D surface, trying to explain that the energetics and the barrier are different. But recently, especially in a paper that appeared this week in PNAS, we kind of got the much more unique answer. So we got the free energy for each direction. First the energy, then free energy, ran Brownian dynamics, and concluded that the only reason for the unidirectionality is not that it's different energy for bending the leg, but because the ADP release step is faster in the right direction. That is, the highest barrier in the process, chemical barrier, is the one that controls everything. Essentially, it's barrier and not exothermicity. We also went to the structure and did some rough electrostatic calculations that show that, in fact, this is the energy of removing the ADP in the two structures, and got preliminary confirmation, but also experimentally it is known. And this is the simulation that we got. Please activate it. Activate the movie, please. This model have more kinks than our previous model, and it does go in one direction. It reproduces the power velocity curves and so on. But the main result was, which is described in this rather long paper, is that having what is called power stroke, which means using kinetic energy of the moving of the arm to move in one direction, does not help at all. This energy is dissipated, and that the reason is relatively simple, that one of the barriers in the cycle in the backward, in the forward direction, is lower than the other. So, okay, we could talk on ion channels, but what we have to finish. So, different people that collaborate with us, this is easier to see. So I want to thank all of them, and thank you for listening.